Hi, today we're going to talk about transcription. It is one of the most fundamental ways to regulate gene expression, and it is necessary for protein production. So today you're going to hear a lot about how that messenger RNA is made. Now here comes the voiceover. Here are the structures of RNA polymerase in the three domains of life: Eubacteria, Archaea, and eukaryotes. Homologous subunits are color-coded and clearly demonstrate the high degree of similarity between all three transcription engines, and in particular, how the archaeal RNA polymerase closely resembles eukaryotic RNA polymerase II. RNA polymerases transcribe all kinds of RNA, including messenger RNA, which encodes protein, ribosomal RNA, which is important in translation, transfer RNA, which is also important in translation, pre-messenger RNA, which is an unspliced version of mRNA, small nuclear RNA, small nucleolar RNA, these SNO RNAs, microRNAs, which are important in gene regulation, small interfering RNAs, which are also important in gene regulation, Pee-wee interacting RNAs, which are important in regulating the expression of transposons, and CRISPR RNAs are part of the bacterial response to foreign DNA. So let me go over these in a little more detail. So we have ribosomal RNA, which is both in bacterial and eukaryotic cells. It functions in the cytoplasm, and it forms the structural and functional components of the ribosome. Messenger RNA is both in bacterial and eukaryotic cells. It functions in the nucleus and cytoplasm, and it carries the genetic code for proteins. Transfer RNA, which is in both bacterial and eukaryotic cells, functions in the cytoplasm and helps incorporate amino acids into the polypeptide chain during translation. Small nuclear RNAs are specific to eukaryotes, and they function in the nucleus, and they're involved in the processing of pre-mRNAs. Small nucleolar RNAs, these SNO RNAs, function in eukaryotes in the nucleus and are important for the processing and assembly of ribosomal RNA. MicroRNAs are in eukaryotic cells. They function in both the nucleus and the cytoplasm, and they inhibit the translation of mRNA. Small interfering RNAs, siRNAs, are in eukaryotic cells, and they function in both the nucleus and the cytoplasm, and they trigger the degradation of other RNA molecules. Pee-wee interacting RNAs, piRNAs, function in eukaryotic cells to suppress the transcription of transposable elements in reproductive cells, which is in the germline. So these are transposons. And CRISPR RNAs are specific to prokaryotic cells, and they assist in the destruction of foreign DNA. And they work a little bit like an adaptive immune system for foreign DNA. RNA structure can be very complex. As they are single-stranded, they can fold into a great number of different secondary structures that can give them a three-dimensional structure similar to a protein. And here I'm going to show you a video now that shows you how RNA can fold.
The nucleic acid is relieved of the constraint of being sort of lockstep with another strand in a double helix. It's free to take on all kinds of shapes because it can interact with other parts of itself. So the structures that RNA can achieve have a wonderful variety, more on the order of what a polypeptide or a protein molecule can achieve. And this is what allows RNA to be a biocatalyst rather than simply an informational molecule.
RNAs are most famous for transcription. RNA synthesis is complementary and anti-parallel to the template strand, as shown here. The RNA transcript has the same base sequence as the non-template strand. New nucleotides are added to the 3' hydroxyl group at the end of the growing RNA strand, and transcription proceeds in a 5' to 3' direction. Transcription can occur on either strand. Here you have 2 genes that are being transcribed from one strand and 1 gene that's being transcribed from another. The double helix, of course, has 2 DNA strands, one positive and one negative. Each gene is transcribed from a single DNA strand, that is complementary to the sequence of the RNA, and that can be either plus or minus. But any given gene may be transcribed from either strand. There are 3 steps of bacterial transcription, and these also apply to eukaryotic transcription. But bacterial transcription is a little less complicated. Initiation. The transcriptional apparatus assembles on the promoter and begins the synthesis of RNA. Elongation. RNA polymerase binds to the DNA and unwinds it. The polymerase then adds nucleotides, one at a time, to the 3' end of the growing RNA strand. Termination. The recognition of the end of the transcriptional unit and the separation of the RNA molecule from the DNA template. The transcriptional unit contains all the DNA necessary to encode an RNA molecule. This includes the promoter region, the transcription start site, the RNA coding region, and transcription terminator sequences. The terms upstream and downstream refer to the direction of transcription and the location of nucleotide sequences surrounding the RNA coding sequence. The RNA polymerase complex is recruited to the DNA, and then RNA synthesis begins. The initiation of RNA synthesis does not require a primer. The RNA is extended and new nucleotides are added to the 3' end of the RNA molecule by the polymerase. The polymerase extends the RNA and unwinds the DNA at the front of the transcription bubble, and then also rewinds the DNA once the transcription bubble has moved through. All of this depends on the RNA polymerase. And here is the bacterial RNA polymerase. It is composed of several subunits, and this is the actual crystal structure. Bacterial RNA polymerase is composed of several factors. Bacterial cells usually have only one type of RNA polymerase, which synthesizes all types of RNA, and that is not true of eukaryotes. The core RNA polymerase contains 5 subunits: 2 copies of alpha, 1 copy of beta, 1 copy of beta prime, and 1 copy of omega. The sigma factor binds to the complex and allows it to bind to the promoter and initiate transcription at the proper start site. So let's see how this works in action. So you have the core RNA polymerase plus the sigma factor, which come together on the promoter. The sigma factor associates with the core enzyme to form a hollow enzyme, and that is the active version of the RNA polymerase. The holo enzyme binds to the minus 35 and minus 10 consensus sequences in the promoter. The template strand unwinds. Then a nucleoside triphosphate complementary to the DNA at the start site serves as the first nucleotide in the RNA molecule. Transcription proceeds, and 2 phosphate groups are cleaved from each subsequent nucleoside triphosphate, creating an RNA nucleotide that is added to the 3' end of the growing RNA molecule. The sigma factor is then released as the RNA polymerase moves beyond the promoter. So RNA transcription is initiated when core RNA polymerase binds to the promoter with the help of the sigma factor. And here's an animation that summarizes prokaryotic transcription. So I'm going to go through this animation and see how I do in placing the different components that are important for bacterial transcription. So here we go.
A transcription unit is a stretch of DNA that encodes a single RNA molecule and the sequences necessary for its transcription. Complete the bacterial transcription unit by placing the remaining critical regions on the DNA duplex at the bottom of the screen.
So this isn't too hard. Let's see here. We got the promoter that goes upstream of the RNA coding region. And then we have terminator sequences within the RNA. Let's see. Yes, okay. And then the telomere and the origin of replication have nothing to do with transcription. They're involved in DNA synthesis and chromosome stability here. All right, let's see what the book tells us.
The 3 critical regions of a transcription unit are the promoter, the RNA coding region, and the terminator. Now, add individual elements to the critical regions of the transcription unit to create a DNA sequence ready for transcription.
Okay, this is a little bit harder, but we know at the end we have a poly A, and this is important for translation, as well as the end of transcription. We have a transcriptional start site right after the promoter. Okay. Then we have some upstream promoter sequences which can be important. Then we have the consensus sequence, which is really important for transcriptional initiation. And those are at minus 35 and minus 10, usually in bacteria. So good, that's right. And then there is an inverted repeat that functions in termination. So here we got this. Okay, got it. The centromere plays no role here. So let's see if I'm right.
Eukaryotic promoter contains minus 35 and minus 10 consensus sequences, and sometimes contains an upstream element. The transcription start site is the first nucleotide of the RNA coding region. The terminator contains an inverted repeat, followed by a stretch of poly A. Unlike DNA synthesis, the initiation of RNA synthesis does not require a primer. Transcription is initiated when the RNA polymerase holoenzyme, composed of the core enzyme and sigma subunit, binds to the promoter. As the holoenzyme unwinds the DNA, a transcription bubble forms, creating a region of single-stranded DNA where transcription can take place. Only one of the 2 DNA strands, the template strand, is generally transcribed. RNA synthesis is complementary to the template strand and is in the 5' to 3' direction. RNA polymerase moves downstream, progressively adding nucleotides to the RNA molecule according to the sequences on the template. Row-independent terminators contain an inverted repeat followed by a poly-A region. When the inverted repeat has been transcribed, it forms an RNA hairpin, which causes RNA polymerase to pause. The stretch of AU base pairs is unstable and causes the RNA strand to separate from the template during the pause. When transcription has ended, the DNA duplex reforms and RNA polymerase dissociates from the DNA.
The basic rules of transcription are these. Transcription is a selective process. Only certain parts of the DNA are transcribed at any one time. RNA is transcribed from single-stranded DNA. Within a gene, only one of the two strands, the template strand, is usually copied into RNA. Ribonucleoside triphosphates are used as a substrate in RNA synthesis. Two phosphate groups are cleaved from a ribonucleoside triphosphate, and the resulting nucleotide is joined at the 3' hydroxyl group of the growing RNA strand. RNA molecules are anti-parallel and complementary to the DNA template strand. Transcription is always in the 5' to 3' direction, meaning that the RNA molecule grows at the 3' end. Transcription depends on RNA polymerase, a complex multimeric enzyme. RNA polymerase consists of a core enzyme which is capable of synthesizing RNA and other subunits that may join transiently to perform additional functions. A sigma factor enables the core enzyme of RNA polymerase to bind to the promoter and initiate transcription. Promoters contain short sequences crucial in the binding of RNA polymerase to DNA. These consensus sequences are interspersed with nucleotides that play no known role in transcription. RNA polymerase binds to DNA at a promoter, begins transcribing at the start site of the gene, and ends transcription after a terminator has been transcribed. Topoisomerase enzymes remove supercoiling that develops ahead and behind the transcription bubble, as the DNA is unwound and rewound during transcription. Now is a good time in the lecture to take a break. Enjoy this Jackson Pollock. Eukaryotic transcription is a little bit more complex. Eukaryotic promoters consist of a core promoter and a regulatory promoter that both contain consensus sequences, as is pictured here. The initiation of transcription requires modification of chromatin structure so that the DNA is accessible to the transcriptional machinery. Transcription factors and RNA polymerase assemble into the basal transcription apparatus, which binds to DNA near the start site and is necessary for a transcription to take place at minimal levels. To get higher levels of transcription, transcriptional activators bind to other consensus sequences in promoters and enhancers and are able to affect the rate of transcription. Here is a schematic of how eukaryotic transcription works. For example, transcription factor 2D binds to the TATA box and the core promoter, and this is a very important sequence for transcriptional initiation in eukaryotes. Then the complex begins to form. The transcription factors and RNA polymerase II bind to the core promoter. Transcription activator proteins bind to sequences in enhancers. the DNA loops out, allowing the proteins that are bound to the enhancer to interact with the basal transcription apparatus. Transcriptional activator proteins bind to sequences in the regulatory promoter and interact with the basal transcription apparatus through the mediator. So as you can see, eukaryotic transcription involves many more components. But still, the most important component is RNA polymerase. There are multiple polymerases in eukaryotes. For example, in animals, there are polymerase 1, 2, and 3. RNA polymerase 2 transcribes genes that encodes proteins, and it is pictured at the left. DNA enters the polymerase through a groove and then unwinds. The DNA-RNA duplex is bent at a right angle, putting the 3' of the RNA at the active site. Nucleotides are then added to the 3' of the RNA. And here's an overview of transcription.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
And here we can see that process in molecular detail.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Once an RNA is transcribed, it is subsequently processed, so it is ready for translation. And this involves things such as splicing and polyadenylation. Here is an overview of this process.
A eukaryotic gene is composed of upstream sequences such as the promoter, the RNA coding region, and downstream sequences. Many RNA coding regions consist of coding regions called exons and non-coding regions called introns. Many genes also contain a 3' consensus sequence. Before an RNA transcript is translated into protein, its pre-mRNA form is extensively processed. The first processing step is the addition of a cap at the 5' end of the pre-mRNA. Capping takes place rapidly after the initiation of transcription. RNA processing continues with polyadenylation. First, the pre-mRNA must be cleaved shortly after the 3' consensus sequence. Then, from 50 to 250 adenine nucleotides are added to the cleavage site. The next step in RNA processing is the removal of introns. An intron contains 3 consensus sequences critical to splicing: a 5' splice site, a branch point, and a 3' splice site. In the first step of splicing, the 5' end of the intron is simultaneously removed from the upstream exon and attached to the branch point. In the second step, the 5' end of the downstream exon is removed from the intron as the exon is spliced to the upstream exon. Splicing continues until all introns are removed. The mature processed transcript is now called messenger RNA or mRNA and contains the 5' untranslated region, the protein coding region, and the 3' untranslated region. The mature and correctly processed mRNA is transported out of the nucleus and into the cytoplasm, where translation by a ribosome can take place. What are the consequences of failed pre-mRNA processing? Drag and drop the X over the 5' cap, the introns, or the 3' consensus sequence if you would like to discover the result of incorrect capping, polyadenylation, or splicing.
So let's look at incorrect splicing.
If splicing fails, 5' capping and polyadenylation will still proceed normally. If splicing is prevented by, for example, mutations at the branch points, then the RNA will be unable to move out of the nucleus into the cytoplasm and will therefore not be translated.
Let's see what happens if the 5' cap is not properly processed.
If 5' capping of pre-mRNA does not take place, splicing, stability, and translation of the RNA are all affected. First, the lack of a 5' cap will affect the removal of introns. Second, mRNA that lacks a 5' cap is less stable and will quickly degrade compared with mRNA that has a 5' cap. Finally, if mRNA lacking a 5' cap is transported into the cytoplasm, the ribosome will not recognize the mRNA, and the mRNA will then degrade.
Let's see what happens if there's problems in polyadenylation.
If the 3' consensus sequence does not function properly, the cleavage of the pre-mRNA cannot take place. The result is mRNA lacking a poly-A tail. Splicing will take place normally in mRNA lacking a poly-A tail. However, an mRNA that lacks a poly-A tail is unstable and quickly degraded by cellular enzymes. Therefore, it will not be available for translation.
Eukaryotic gene expression is complex, and it can be regulated at many steps, including both transcription and translation. So this overview is a bridge between what we've learned today about transcription and what we'll discuss in the next lecture, which is on translation. I hope you find it helpful.
How is genetic information passed from genotype to phenotype? In eukaryotes, 3 processes convert the information found in DNA to protein: transcription, RNA processing, and translation. Transcription, the first step in gene expression, uses DNA as a template. The DNA of a gene contains a promoter and an RNA coding region. The RNA polymerase transcription machinery binds to the DNA at the promoter and synthesizes a pre-mRNA as it moves along the RNA coding region. A 5' cap is added to the RNA shortly after transcription starts. The pre-mRNA must be processed before it can be translated into protein. The 3' end of the pre-mRNA is cleaved and a poly-A tail is added. Many eukaryotic genes are split into exons and introns. A spliceosome removes each intron, resulting in a mature mRNA. The mature mRNA is transported to the cytoplasm for translation, the final process of gene expression. Proteins that bind to the poly-A tail interact with proteins at the 5' cap, causing the mRNA to form a circular structure. These proteins initiate the assembly of the ribosome near the 5' cap. Protein synthesis begins at the translation start site. One mRNA molecule is translated by multiple ribosomes, resulting in many copies of the protein. Translation completes the process of gene expression. Information from DNA has been passed through an mRNA to produce a functional protein. Different types of DNA mutations can alter the process of gene expression in different ways. Choose a mutation to find out how the flow of genetic information is affected. This animation follows what happens to a mutation in the coding region of a gene through the process of gene expression. First, the DNA is transcribed. Because the pre-mRNA sequence is determined by the DNA sequence, the mutation is passed on to the pre-mRNA. Next, the pre-mRNA is processed. A poly-A tail is added to the pre-mRNA, and the introns are spliced out. Because the mutation is in a coding region in the third exon, it ends up in the mature mRNA. Using the mRNA as a template, ribosomes synthesize each protein one amino acid at a time. The presence of the mutation in the mRNA leads to the incorporation of an incorrect amino acid. In this case, the result is a mutant protein with an altered shape. The DNA mutation has been carried through the steps of gene expression to the protein. Because the mutant protein is misfolded, it is likely to be non-functional. This animation describes what happens if a gene contains a mutation in its promoter region. The promoter of this gene contains a mutation at a critical site for RNA polymerase binding. The polymerase and the rest of the transcription machinery fail to bind to the DNA and carry out transcription. Because transcription is blocked, no mRNA is synthesized, and therefore no protein is made. The mutant gene is not expressed. The effect of an intron mutation will be evident at the RNA processing step of gene expression. First, the DNA is transcribed. Because the pre-mRNA sequence is determined by the DNA sequence, the mutation is passed on to the pre-mRNA. Next, the pre-mRNA is processed. A poly-A tail is added to the pre-mRNA, and the introns are spliced out. The intron mutation is not at any sites critical for splicing, such as a branch point, and so splicing takes place normally. The mutation is removed with the discarded intron and is thus not present in the mature mRNA. The wild-type mRNA is translated into a normal protein. The intron mutation has been spliced out of the mRNA in the course of RNA processing. Thus, the mature mRNA and resulting protein are wild type. This type of mutation does not affect the functional product of gene expression. A nonsense mutation changes a codon that normally specifies an amino acid into a stop or nonsense codon. This change affects the translation step of gene expression. First, the DNA is transcribed. The mutation lies in the RNA coding region, and so it will be present in the pre-mRNA. Next, the pre-mRNA is processed. A poly-A tail is added to the pre-mRNA, and the introns are spliced out. Because the mutation is in a coding region in the third exon, it ends up in the mature mRNA. Using the mRNA as a template, ribosomes synthesize each protein one amino acid at a time. The nonsense mutation is read as a stop codon, prematurely terminating translation. A truncated protein is released. The nonsense mutation has been carried through to the mature mRNA, causing the synthesis of a truncated protein. This mutant protein is likely to be non-functional.
Here are some genetic terms and concepts that I think you should know. Messenger RNA, an RNA molecule that carries genetic information for the amino acid sequence of a protein. Template strand, the strand of DNA that is used as a template during transcription. The RNA synthesized during transcription is complementary and anti-parallel to the template strand. Transcription unit. Sequence of nucleotides in DNA that encode a single RNA molecule, along with the sequences necessary for its transcription. A transcription unit normally contains a promoter, an RNA coding sequence, and a terminator. Promoter. DNA sequence to which transcription apparatus binds so as to initiate transcription. Indicates the direction of transcription, which of the two strands is to be read as the template, and the starting point of transcription. Terminator. Sequence of DNA nucleotides that causes the termination of transcription. RNA polymerase. Enzyme that synthesizes RNA from a DNA template during transcription. Sigma factor. Subunit of bacterial RNA polymerase that allows the RNA polymerase to recognize a promoter and initiate transcription. Transcriptional activator protein. Protein in eukaryotic cells that binds to consensus sequences in regulatory promoters or enhancers and affects transcription initiation by stimulating the assembly of the basal transcription apparatus. General transcription factor. Protein that binds to eukaryotic promoters near the start site and is a part of the basal transcription apparatus that initiates transcription. Tata box. Consensus sequence, TATAAAA, commonly found in eukaryotic RNA polymerase II promoters, usually located from 25 to 30 base pairs upstream of the transcription start site. The Tata box determines the start point for transcription in eukaryotes. Core promoter, located immediately upstream of the eukaryotic promoter. DNA sequences to which the basal transcription apparatus binds. Regulatory promoter, DNA sequence located immediately upstream of the core promoter that affects transcription. Contains consensus sequences to which transcriptional regulator proteins bind. Enhancer. Sequence that stimulates the maximal transcription of distant genes. Affects only genes on the same DNA molecule, therefore is cis-acting, not trans-acting. Contains short consensus sequences. Is not fixed in relation to the transcription start site. And can stimulate almost any promoter in its vicinity. It also may be upstream or downstream of the gene. So basically, it's very difficult to find an enhancer, but they can have a profound effect on transcription. Well, I hope you've enjoyed the lecture. Next time we will discuss translation. See you then.